Well, welcome. I get to welcome you all to the Myrtle Wilson Performing Arts Center here at Gonzaga, and to those of you on Zoom to our Spring 2024 Next Generation Medicine Lecture. We're excited to have Dr. Jeff Iliff with us from the University of Washington School of Medicine, here to share his compelling research on how sleep affects brain wellness. I am Dr. Julie Walter. I am the new dean for the School of Health Sciences over here at Gonzaga University in partnership with University of Washington Medical School. And the University of Washington School of Medicine and Gonzaga University formed a health partnership in 2016 with a vision to improve the health and prosperity throughout Eastern Washington by transforming medical education, research, and innovation. And our combined strengths of each institution, deep community roots and world-class health and medical education, and our vast networks of students, faculty, healthcare professionals is what's really creating an impact in preparing the next generation of healthcare professionals to advance discovery and development in new ways to care for patients. And so it's just an opportunity to tell you a little bit about some of the updates of our collaborations as more people are coming in and finding spaces. We at the UW-GU partnership are finding ways to collaborate across our institutions, but with also our different disciplines. So for example, we have co-appointed students, our human physiology professors at Gonzaga who teach undergraduates in human physiology are also teaching our med students in their anatomy physiology labs. We have a new department of public health at Gonzaga University where we have co-appointed faculty that now work with capstone research projects for our medical students that will also include in the future, starting in the fall, our undergraduate students studying public health at Gonzaga. So those are just some examples of cross collaborations as we think about our interdisciplinary education with our partnership. So tonight's lecture is another example of how we're sharing the proven excellence of our two institutions and how we're showcasing the vast regional network of partners who are delivering medical excellence. Together, we've created a solid foundation of collaboration that makes us stronger and better together. So with that, I'd like to turn to our lecture for tonight. Our Next Generation Medicine lecture series is presented to the Spokane region 3 times a year and features some of University of Washington School of Medicine's most highly respected educators and researchers to share discoveries and insights about public health topics. We welcome you to Gonzaga and hope you take away some new knowledge tonight. And now I'd like to introduce my colleague, Dr. Daryl Podick, another fellow dean over at University of Washington, who will say a few words and introduce Dr. Iliff.
Thanks, Julie. And thanks to all of you for coming tonight. It's a beautiful spring night out there, and you're indoors here listening. So we really appreciate that. It's going to be a great talk tonight. Dr. Iliff is well-renowned, and he's going to provide us with some great information. So as Julie said, I am the Associate Dean for the University of Washington School of Medicine. I oversee all of our activities here in Eastern Washington, and I'm Chief for Medical Education for our UW-GU partnership. We do have some sponsors tonight who I'd like to thank before we get started. Those include the Spokane Regional Health District, the Spokane County Medical Society, and the University of Washington Public Health Initiative, the Population Health Initiative. What I'd like to do is share a little bit of how things are going to flow this evening. Dr. Iliff will give his presentation for about 45-50 minutes. And then we're going to ask Dr. Don Howard, who's a local sleep doctor here in Spokane, to come join us for your questions and hopefully their answers. So if you have questions, we're going to have some mics. You can save up your questions for the end of the presentation, and we'll try to make sure we get as many answered as we can. So it is my pleasure to introduce Dr. Iliff tonight. He comes from University of Washington. He's been to Spokane before, so we don't have to do too much of that introduction. Jeff received his undergraduate degree in zoology from the University of Washington. Then he went and pursued his Ph.D. in physiology and pharmacology at OHSU in Portland. He's a professor in various departments at the University of Washington, primarily in the Department of Psychiatry and Behavioral Health Sciences, and also in neurology. He's the associate director, as you can see, of the MIRECC, the Mental Illness Research, Education, and Clinical Center at the VA in Puget Sound. And his research focuses on neurodegeneration of the brain, traumatic brain injury, and he is involved in the UW Alzheimer's Research Center. He's here tonight to talk to us about how sleep impacts the health of our brain, and in particular, how he is working on the glymphatic system, which is something newly described and coming out of his lab. And look forward to him talking to us about brain health and sleep and the connection. And I know you're here to listen to him talk, not me. So, Jeff, come on up.
Great. Thank you, Daryl, for that kind introduction. Thank you to the University of Washington for bringing me here, to Gonzaga for hosting us in this fabulous venue. So over the next 50 minutes or so, 45 minutes, depends on how fast I talk, I'm going to be walking with you through some science that has emerged over the last dozen years or so. So all of this started back in 2012. That has shed a good deal of light on how we understand the relationship between aging, sleep, and the development of dementing disorders like Alzheimer's disease. So we're gonna talk about where the science was and where we're going with that right now. I have a couple of mandatory disclosures. So the two that are important is I do serve as the chair for the scientific advisory board for a startup company called Applied Cognition. They compensate me and I have equity in that company. I won't be talking about what they do at all in this talk, so that shouldn't be relevant. I'm also a full-time federal employee, and all of these views that you will hear are mine and not those of the federal government. I don't know whether they or I are more happy for that. I don't know how that goes. A couple of other things. So I want to just set some expectations. This is a science talk. I am a person who believes that the public should hear science, and they shouldn't necessarily hear it through the filter of media sources or, heaven forbid, from the internet. I think the public can and should hear and understand things about their bodies and their health. And it's my job as a researcher to be able to describe and explain these things in a way that a normal human can actually understand. I hope that you'll follow with me. I will be showing data. I will be showing rodent data, human data. There will be plots. I promise there will be no math. So I'm not a math person, so don't worry about that. If something doesn't make sense, it's probably my fault, and you can complain about it at the end. So tonight, we're going to be focusing on talking a lot about three big topics. We're going to be talking about the brain. We're going to be talking about sleep. We're going to be talking about Alzheimer's disease. I don't really know how much about those three things anyone in this room knows, so we're going to cover a little bit of basics at the very beginning just to make sure that we're all on the same page. So let's talk about the brain first. Brain is a computational machine. It's a high-performance machine. The human brain is incredibly complex. So there are tens of billions of electrical cells within the brain that form hundreds of trillions of synapses. Those are the connections between those electrical cells. Those are electrically firing really all the time, such that there are billions to trillions of electrical impulses happening in your brain every single second, right? So it's a huge amount of electrical activity. All of that electrical activity is the basis. It's the biological basis for everything that your brain does, for your hearing, for your smelling, for your feeling, for your memory, for your motion, for the way that you make decisions. All of it comes down at a wiring level to the firing of these neurons and their very, very intense electrical activity. That intense activity comes at a massive metabolic cost. So the brain receives a wildly disproportionate amount of the body's blood flow to supply this metabolic need. So this is a corrosion cast of the human brain's vasculature. So what they did is they took cadaver, they filled the blood vessels with polymer, and then they used a chemical to erode away everything that isn't plastic, and what you're left with is the brain's vascular network. And what you can appreciate is its shocking complexity, right? Such that essentially every single one of those neurons, of those tens of billions of neurons that I talked about, every single one of them almost has its own capillary, which is the smallest blood vessel in the brain, right? So it's this very active, intense, high-performance organ that requires a lot of energy, but also produces a lot of waste, which we'll talk about a little bit later. Sleep. Sleep is something you all do, I hope. I hope you did it last night. I hope you will do it tonight. I hope you won't do it in the next 45 minutes. If you do, good on you. So sleep isn't one thing. Sleep is it's a complex behavior. While your body may be resting, but your body's doing things during sleep, your brain is actually doing a lot of things. So when you look at the brain and what it's doing during sleep, what you can observe is it's doing several different electrophysiologically distinct things, right? So it's passing into and out of these different stages from things like deep sleep, which is characterized by these very slow, synchronous oscillations of electrical charge, right? To very rapid asynchronous activity that's happening during REM. So this is the phase of sleep that's most commonly associated with dreams. And through the course of the night, normal people who are healthy will pass through a number of sleep cycles during which they'll go down into deep sleep, up into REM, back down, back up. They got up to go to the bathroom, and then they deep sleep, and so on. And through the course of the night, you'll typically go through 3 to 5 of these cycles. The cycles actually change in their composition. Early in the night, they tend to be heavy in this slow wave sleep, and toward the end of the night, they tend to be heavier in REM sleep, right? So this is normal sleep. The take-home message from this slide, though, is that sleep, your brain isn't resting, it's doing something. And this electrical activity probably has a purpose, and probably has an important purpose. Alzheimer's disease. Alzheimer's disease is something everyone is thinking about. Many of us have loved ones or friends or family with dementia. My mom has vascular dementia right now. I'm sure many of your parents or aunts and uncles or folks in your life also have dementia. Alzheimer's disease and dementia are not synonyms. Dementia is a condition. Alzheimer's disease is the most common cause of dementia. There are other causes of dementia, like vascular dementia, normal pressure hydrocephalus. You can become demented from problems with your thyroid hormone, right? Alzheimer's disease, to have Alzheimer's disease, you have to have 2 things. And those are buildups of different proteins in your brain. So there's buildups of a protein called amyloid beta in plaques in the spaces between your brain cells. And then there's buildups of another protein called tau inside of neurons. So these amyloid plaques and these tau neurofibrillary tangles is what these are called. These are the hallmarks of Alzheimer's disease. If someone dies with Alzheimer's disease, historically, the way that you knew it was they were demented, and then at autopsy, they look for this, and when they see it, they say, that person had Alzheimer's disease, right? So this is a graph. So if you give an Alzheimer's research talk in the United States, you are required by law to show this graph. That's not actually true, but it is almost true. What this graph shows is what is generally accepted to be the sequence of events that ends in dementia caused by Alzheimer's disease, right? One of the things that you can appreciate is if we look all the way on the right here, the last thing that happens in this series of events is cognitive and functional decline. So this is the thing. This is where you take your mother to the physician and say, she's forgetting stuff, she's making bad decisions, she's losing her way, she's actually having trouble doing day-to-day life, right? So that's only at the very end of the process. But a lot of things are happening before that, and I'll draw your attention over here, way to the left. So this is the first development of these amyloid plaques here with this red line. This happens way before cognitive impairment. This is the development of these tau tangles. That happens after it. This starts happening decades before a person winds up at the memory clinic with cognitive impairment. So the process that is believed to underlie Alzheimer's disease is actually latent. It's burning in people's brains for years and years before the onset of symptoms that would lead a person to start worrying about cognitive impairment and dementia. What that tells us is that if we want to not just treat Alzheimer's disease, if we want to prevent Alzheimer's disease, we need to get upstream. We need to get over to the left. So you're probably familiar, you probably know, you've read in the news that there's a couple of new drugs out there that are antibodies that target amyloid, right? It targets this red thing, these clumps. They do a pretty good job of removing them, right? So if the pathology is there, you have to do a PET scan or do a lumbar puncture to measure if you have these plaques. And then if you do, they can now start to give you these antibodies that'll get rid of these plaques. And the hope is, by getting rid of them, you can prevent this cognitive impairment. This is what's called secondary prevention, meaning the pathology is there, you get rid of it, and you hope to stop progression, right? But you don't prevent the pathology, you're just preventing the consequences of pathology. In heart disease, we strive for primary prevention, right? The idea is that I'm 45 years old. I'm 45 years old, and so now my doctor is starting to pay attention to my blood lipids, and he's starting to pay attention to my blood pressure. Because if I have hyperlipidemia, if I have hypertension, I may not yet have heart disease. But if they treat the hypertension, if they treat the hyperlipidemia with Lipitor or whatever, they can actually prevent the vascular dysfunction that will eventually lead to a heart attack and a stroke. That's primary prevention. So the idea is, if we can identify the processes upstream of amyloid and tau, we actually may be able to identify people at risk of these things and preventing the development of this pathology and the start of this process. That's the ultimate, I think, when you talk to people who are in the Alzheimer's space, that's where the goal actually is. We don't just want to be treating people who already have plaques. We want to be preventing those things from happening in the future. All right, so let's talk about sleep and neurodegeneration. So neurodegeneration is a general term for what's happening in the brain in conditions like Alzheimer's disease or Parkinson's or Lewy body dementia or chronic traumatic encephalopathy, which you've heard a lot about from the NFL players, right? There's a long appreciated relationship between sleep disruption and aging generally and neurodegeneration in particular. So we know, probably from our own experience, I'm starting to notice it myself, I sleep more poorly and less as I get older, right? And I'm sure many of you have noticed the same thing. There's a lot of reasons for that. People who move into cognitive impairment and dementia, they have profoundly disrupted sleep cycles. And in fact, it's disruption of the normal sleep cycle that's frequently the thing that leads a person to have to move from an in-home care environment into an institutional care environment, because you just can't run around after your dad, all through the night while you're trying to sleep, right? So that association has long been known. So I'm going to show you just a quick paper that we published recently that gets at that association, and then I'm going to talk about what the nature of that association might be, why that connection's there. So this is a study that we published it this year. This was run out of the Seattle Longitudinal Study. So this is a big study out of Seattle. That's the name. It started actually in the 1950s, which is crazy to me that there's been a study running that long. And it was run by one person who recently passed away. So the interesting thing about the study is it's been following people in the Seattle area for decades and decades and decades. And sometime in the 90s, they started asking people about their sleep. They started saying, about how much do you sleep every night? And people would answer. And so what they have is they have between 10 and 20 years of sleep data on people, right? So we have all the sleep data on hundreds and hundreds of these participants. And so we looked at how their sleep behavior related to whether or not they would develop cognitive impairment later on. And we saw two main things. The first is that's this line here. So this is, if one of these people was a short sleeper, and the short sleeper in this case was defined as less than 7 hours a night, they exhibited an earlier decline in cognitive function. This is a survival curve that lists your survival with intact cognition, right? So dropping off means you have cognitive impairment. So short sleepers have earlier cognitive impairment. That's not new. We've actually, a number of studies have reported this. That is entirely not novel. The interesting thing that we did see is because we had a bunch of measurements from the same people over and over and over again, we were able to look at trajectories of how sleep was. And it turns out that what we saw was that people who exhibited the greatest variability in sleep, so 1 year they would say 9 hours, 1 year they'd say 6 hours, and 1 year they'd say 9 hours, and 1 year they'd say 5 hours, right? So people with variable sleep patterns, the more variable, the higher the risk of cognitive impairment, suggesting that long-term patterns of sleep are associated with cognitive impairment. Important caveat. When people think about the association between poor sleep and dementia, the assumption had always been, well, as the brain degenerates, the part of the brain that controls sleep degenerates. And that's why sleep gets all screwed up, right? It's because the part that's supposed to regulate sleep isn't working properly, right? That's probably true. That's probably why in people with dementia, you see really profoundly disrupted sleep. But these people aren't cognitively impaired at all, right? When we were measuring their sleep, we were measuring their sleep 10, 15, 20 years before cognitive impairment, right? So the sleep before cognitive impairment was dictating how cognitive impairment was going to happen. This is another study, and there's actually been several studies just like this, but this is a good one because it has pretty pictures. This is published in 2013 out of Johns Hopkins in Baltimore. This is a study in cognitively intact older people, so people over the age of 65, but who do not have cognitive impairment. So these are healthy cognitively intact individuals. They used an imaging approach called positron emission tomography to measure the burden of these plaques in the brain. So they can use a radio tracer to measure how much it is. And in these things, cold colors are low, hot colors are high. And they asked the people, how much do you sleep and how well do you sleep? And in general, the people who reported sleeping less than 6 hours a night, you can just look, lots of red, not as much red, had more amyloid burden. And this was also true of people who reported sleeping poorly. So not just short sleepers, but also people who just say, man, my sleep is bad, right? It's low quality. So with these data suggest that short sleep and poor sleep is actually promoting the development of this pathology that underlies Alzheimer's disease. And this is before cognitive impairment. This is in those decades before. So what these data start to suggest is maybe sleep or its absence is promoting the development of this pathology. It's actually driving the bus instead of following the bus. This is another study. I'm gonna show you study after study after study. So there's gonna be a lot of data. So just be ready for that. This is a crazy study. I don't know who they got to do this, but good for them. So essentially, this was done at Washington University, the other Washington University. This one's in St. Louis, not the University of Washington, which is very confusing. They took older individuals. They put an indwelling catheter in the lumbar intrathecal space. So the same place where you would get a spinal tap or kind of near where you would get an epidural, right? And they sampled the fluid from around the brain every hour for 24 hours. And they took that fluid and they measured how much amyloid is in it every hour. And they plotted it. This was a study that actually, this was published about 15 years ago. They did it in mice and in humans both. This is the human data. So what it showed was that during the day, human amyloid levels, so remember that's the thing in the plaques, goes up during the day and declines during the night. So in a normal healthy individual, amyloid levels are actually, they're being regulated by the sleep-wake cycle. If you they actually did this experiment where they took these people and they sleep deprived them overnight, and this drop that's supposed to happen doesn't happen. So while we think about amyloid and the buildup of these proteins as something that is happening over the time scale of years or decades, this data suggests that there's a much tighter connection between sleep and what this stuff's doing in your brain through the course of a day or 2 days or 3 days. So what is that relationship? So take a pause. If you've lost track, this is what we just talked about. Emerging data suggests that sleep disruption, particularly midlife, appears to be promoting the development of dementing disorders like Alzheimer's disease. But the question is why? What is the process that links sleep with the development of this pathology? And that's the work that our group has been doing over the last 10 or 12 years. So let's talk about brainwashing. Not the cult brainwashing, the other kind. So I mentioned a couple slides back, all the neurons, all the blood vessels, high metabolic burden, lots of electrical activity, brain's producing a lot of waste. Beginning in around 1950 and up until about 2010, the way that people thought that the brain got rid of waste was through the fluid that surrounds the brain. So the brain rests in a pool of this cerebral spinal fluid, which is this protein-free, cell-free fluid. It actually floats the brain, so it helps support the brain. It cushions the brain, right? That's mostly what people think about it. But going back into about 1950 or so, the thinking was that waste in the brain that couldn't clear to the blood, a lot of stuff goes out into the blood, like carbon dioxide is a waste. It goes out into the blood, right? Some things can do that, but a lot of things can't. Amyloid beta is one of the things that can't necessarily go out to the blood. Tau is another thing like that. The idea was that what it did is it sort of percolated its way out to the fluid that was outside the brain, or percolated its way down into the fluid that's inside the brain. And then as this fluid turned over, that's how the brain got rid of its waste. So beginning in 2010 and for the next several years, we started to ask the question, is this fluid just sitting out here, or does it enjoy a more active and more exciting life than that? Is it doing something more? So this is a study in mice. This is an approach that we called in vivo two-photon microscopy. In vivo means in a living animal. So this is imaging into the brain of a living mouse through its skull, right? It's a simple experiment. We've done two things. We've labeled the blood vessels with a red tracer. So these are blood vessels down inside the brain. There's blood vessels here at the surface of the brain. They're kind of washed out now. And then we injected a green tracer into the fluid around the brain, and we just watched over time what happened. And the interesting thing that we saw was that green tracer, it didn't just stay on the surface of the brain, right? And it didn't move vaguely over the surface of the brain, very diffusely, but rather it followed very specific anatomical pathways. Here, so you notice it arrives along this blood vessel. And then as this blood vessel turns down through the brain, through the cortical mantle, you can see that green tracer follows it. And as we keep going down, you can see that there's more green tracer following it. And as you move down, there's more green tracer following it. And so this green haze that you see is actually fluid washing through brain tissue. So it's not just sitting on the outside, but rather it's washing through the spaces between the brain cells. It's actually kind of interesting the way it does it. So if we zoom into one of these blood vessels, so the blood is labeled in red, the CSF, the fluid, is labeled in green. And so the fluid isn't in the blood vessels. It's around the outsides. And so there's these little channels, they're called perivascular, peri meaning around, perivascular spaces that serve as conduits for the movement of this fluid. So essentially the brain is repurposing its blood vasculature. So remember how complex that was? You had all those vessels going all through the brain. It actually uses that as a structural scaffold to permit fluid to wash through brain tissue. That's how the brain is actually clearing out some of its waste. It's not something that's happening at the same rate all the time. So this is a similar experiment. Here what I'm showing you is down inside the cortex, so inside the brain of a mouse. This is actually the same mouse twice, right? So the blood vessels are labeled in blue. This mouse is first, it's awake, and then it's sleeping. And you might think, oh, poor guy, his video's not working. But it's actually working, it's just that not much is happening. What's happening is if we measure how much of that fluid is, or how much of that tracer is coming through, in the wake state, almost none is. Whereas when the animal shifts into a sleeping state, its situation completely changes. So the brain, at least the mouse brain, when it goes to sleep, is actually shifting into a cleaning mode that it isn't in during its normal waking life. And this isn't just the movement of fluid into the brain, this is actually the clearance of things out of the brain, right? So this is an experiment where if we inject stuff, right, so different solutes into brain tissue, so we can inject a labeled version of amyloid beta into brain tissue. We can measure how fast it gets cleared out over the course of a few hours. We can convert these curves into simple numbers so that you can kind of make an easy comparison. And what you see is in sleeping animals, the rate of clearance of amyloid, which is the thing that clumps up in the brains of people with Alzheimer's disease, it's about double what it is in sleep compared to waking. So during sleep, your brain is actually clearing away, or not your brain, this mouse's brain. This mouse's brain is clearing away amyloid during sleep. So this process, this system of perivascular networks, in 2012, we made up a word because we got tired of saying the perivascular network for fluid exchange through the brain, because that doesn't sound very good. And so we made up this word, this glymphatic system. So GL for glia, so the support cells in the brain, those are things that we think are playing an important role. And lymphatic, because this process of waste clearance from the spaces between cells, that's a lymphatic process. Your brain doesn't have lymphatic vessels in it. It has lymphatic vessels around it, but not inside it. So in your muscles, in your kidneys, in other parts of your body, it's your lymphatic system that does this clearing. But the brain, because it doesn't have lymphatic vessels in there, and there's reasons why that's the case, it seems to serve this lymphatic function by washing fluid through along the outsides of blood vessels. But this is all mice and rats. And to be honest, like, mice and rats are cool. But if this was only a rodent phenomenon, because rodents are weird. Rodents are very strange. There's a reason why we chase them out of our houses. If this was only a mouse or rat thing, you shouldn't care. Maybe professors at universities would care because they care about weird things. But the question is this something that's happening in the human brain? So is this more than just mice? How do we evaluate this in humans? Well, to start with, we're not going to do 2-photon microscopy in a human. There's technical reasons why we can't do that, and there's ethical reasons why we can't do that. So that's out. So we have to find a different way to study this process in people in a way that's going to be suitable and acceptable. You'll notice this isn't a human brain, but I promise I'm getting there. This is a rat. This is a rat MRI study. So down in Oregon, when I was there, I was at Oregon Health Science University for 6 years before coming to the UW. We had a small animal. It was a big machine, but it was an MRI magnet for rats and mice, believe it or not. It cost much less than the MRI that you get. So the experiment that we did it's very similar to what I showed you before. The difference is, instead of using a 2-photon microscope, we're using an MRI scanner. Instead of injecting a fluorescent tracer into the fluid around the brain, we inject in an MRI contrast agent into the fluid around the brain. And then what I've done here is I've shown where the tracer went in the first hour in blue, in the second hour in green, and the third hour in red, just so you can get a little bit of a time course. The cool thing about this is MRI gives you a view of not just a tiny part of the brain, but actually the whole brain all at once. And so what you can appreciate is, in blue, first the CSF tracer, it moves through the CSF compartments at the base of the brain. And then in green, its earliest movement into the brain. You see how it follows a line right there? Well, that's the anterior cerebral artery. You see how it follows a line right there? That's the choroidal artery. See how it follows this line here? That's the posterior cerebral artery. So what we see is, again, the movement of fluid through the entire rodent brain along the axis of these blood vessels. So we're seeing the same thing, but using MRI. The cool thing about MRI is I can do an MRI in a person. I can't do this because only people in Norway do this. So there's a group in Oslo, at the University of Oslo in Norway, who is injecting gadolinium-based contrast agents into the CSF of patients. They're doing it for imaging. These are people who are being evaluated for neurosurgical procedures, right? So they need really good imaging to see what's going on. So there's a reason why they're doing this. So they're not just being ridiculous. But they're using exactly the same approach that we did here in rats in humans. And what they see is that an hour after they injected the contrast agent into the CSF, contrast agent on this MRI shows up white or light. You see it fills the CSF spaces at the base of the brain. You see it starts to enhance the front part of the brain, and it really starts to enhance. And then when participants come back the next day, you see how the brain is lighter? That's contrast agent that's enhancing the entire brain. So that's this washing of contrast. They're seeing it in people. So they're able to see the very same washing process in people using contrast-enhanced MRI that we saw in rodents. Well, what about sleep? I think this was one of the most important papers to be published in the last couple of years. This was the first paper that validated this sleep washing in humans. So these are the crazy people in Norway again. The reason why we don't do this is because these contrast agents, putting them into you can put them into blood. Lots of people have contrast enhanced MRI that way. Putting them into the CSF is an off-label use, meaning it's not FDA approved, right? You can still do it. A clinician can still make a decision to do that, but it isn't something that's FDA approved. So that's why people don't do this in the US. So what they did is they brought their participants in the afternoon and they injected contrast agent. And then sometime in the evening, they took the people and they divided them into two groups and they measured how much contrast had gotten in. And then between the two groups in the evening, the amount of contrast loading was about the same. And then one of the groups, the blue group, was allowed to sleep normally overnight, and then the red group was kept up all night. And then they were brought back the next day to measure how much of that contrast agent had gotten out of the brain. And what they saw was that the people who were allowed to sleep normally, they cleared the contrast, whereas the people who were sleep deprived, they cleared the contrast less effectively. Even when the people came back the next day, 2 days later, so everyone was allowed to sleep here, that deficit was still there. So it didn't even catch up after a full night's sleep. So what this data shows is this sleep active brain waste clearance is actually happening in the human brain. I've mentioned before already that amyloid beta, which clumps up in the brains of people with Alzheimer's disease, tau, which clumps up in neurons of people with Alzheimer's disease, those are things that are cleared through this process. I didn't show you that data, just take my word for it for now. The question is, does a failure of this process, is this one of the things that could underlie the connection between poor sleep and Alzheimer's? I told you that there's a well-established connection, but what it is, why it is, that's something we don't know. And so that's really the question that we're now trying to get at. So, does slowing brain waste clearance, does it contribute to Alzheimer's disease? I'm going to back up, and I want to talk a little bit about how a scientist proves something. Because sometimes you'll read it, if you go on your news feed and you'll be like, oh, there's a new Alzheimer's treatment out there. I read about it, and then you read carefully, and it was one study in mice that's never been replicated and probably will never be replicated, and it gets kind of crazy. So the question is, if this process is at the root of the connection between sleep and Alzheimer's disease, that's really important. And so how do we actually know if that's true, right? So if we think about Alzheimer's disease or dementia let's think of it as a crime, and we can view this brainwashing as a suspect in the crime, right? What do we need to know to be able to have confidence that this is the suspect that's doing it, and that this is the one that we need to target and to take care of to prevent this thing from happening, right? Well, for me as a scientist, I need a couple of things. First, I need to know that the suspect is at the scene of the crime, right? So the question is, do I see impaired function of this process in settings associated with Alzheimer's disease, right? So in settings of aging or cerebrovascular disease or traumatic brain injury, all of which are risk factors for Alzheimer's disease, do I see impairment of this process? Is it there? Second, does it have the motive and the means? Meaning, in an experimental setting, if I disable this system, can I make Alzheimer's pathology? Right? Is it capable of doing it? Right? Because if it's not capable of doing it, it doesn't do it. Right? If it's not changing, it doesn't do it. These are two steps. Now, it could be at the scene of the crime, and it could be capable of making the pathology or promoting the pathology, but it might not actually be the real thing. It might just be a bystander, or it might be some sort of secondary thing. And so then there's the smoking gun. Do we see evidence that if I do something to change this function, if I enhance it, can I prevent Alzheimer's pathology? Can I prevent cognitive impairment and dementia, right? Because if I could, bingo, right? That's the proof. That's the proof that you need. So we're going to walk through these 3 steps. Spoiler alert, we don't have them all checked off. This is a story in progress, right? But this is how science works. You start with the discovery, and you have to start working through step by step how these things go together. So let's think about the scene of the crime, okay? Neurodegenerative disorders generally, and Alzheimer's disease particularly, are characterized by protein misaggregation, right? So that is the clumping together of proteins that shouldn't clump together. In Alzheimer's disease, we talked about plaques made of amyloid beta. We talked about neurofibrillary tangles made of tau. Those are the hallmarks of Alzheimer's disease. We also see aggregation in other conditions, right? So Parkinson's disease and Lewy body dementia, you see the aggregation of a different protein called alpha-synuclein, right? You see tau aggregate in other conditions that aren't Alzheimer's disease, right? So chronic traumatic encephalopathy, right? Which is the dementing disorder that comes from repetitive head impacts that former NFL players sometimes get. That's a tau pathology. So in common among these conditions is protein misaggregation. Also in common among them is the influence of different risk factors. The risk factors are things that make you more likely to get it, right? And we think that many of these risk factors, what they're doing is they're promoting these processes. Aging. Aging is the biggest risk factor. It's such a big risk factor that you don't even think of it as a risk factor, right? But these are all conditions of the aging brain. Aging is the strongest risk factor. Cerebrovascular disease is a risk factor for these conditions. Exposure to traumatic brain injury, chronic sleep disruption, something we talked about before. These are risk factors for dementing disorder, including Alzheimer's disease. So the question is this brain clearance, is it impaired in these settings? The answer is yes, at least in the animals and in some human studies. I'm going to show you one research vignette. I'm going to walk through one set of data. It's not all there is. If this is something that's of interest to you, you can go to this QR code. It will take you to a list of papers that actually underlie this statement, because you should never believe a scientist when he's talking about his own work. Don't take my word for it. Go check it out if it's of interest to you. But I can tell you that at least in the animal models of aging and cerebrovascular injury and traumatic brain injury and acute sleep deprivation, we do see impairment of this brain clearance. So in the non-genetic risk factors, in animal models of those situations, we do see an impairment. So it does seem to be at the scene of the crime. This is an example. This is a very simple study that we published a few years back. This is asking the question, when animals get old, do they have slower brain clearance? There's nothing wrong with these animals. They don't have a disease. They're just old, right? And being old it's not a disease. It's just something that happens to you, right? These are healthy young adult mice. So these are mice in unhealthy relationships, making bad decisions. They're 18, 20 years old. These are mice typically in a lab setting will live about 2 years or so. So these are pretty old mice, right? So they're doing a lot of golf and maybe collecting seashells or something. But if you measure the movement of tracer into brain tissue in a healthy young animal, you can see it goes through the whole brain. It's very active. In old animals, nothing wrong with them. They're just old. It's slower. If we measure the clearance of amyloid over time, so what we can do is we can measure how much of this amyloid gets cleared in the first 60 minutes. We see that in healthy young animals, 70% of the amyloid is cleared in the first 60 minutes. In middle-aged mice, it's slower. In old mice, it's slower still. So aging is is the strongest risk factor for dementing disorders, and we do see an impairment of this process in aging animals. So check. It does seem to be, at least in the animal models and in some studies in humans, at the scene of the crime. Does it have the means? If I impair this, can I actually make pathology, or can I drive pathology? Can I make it worse? Well, as we talked about before, these conditions are associated with aggregation of amyloid, tau, and alpha-synuclein. Again, don't take my word for this. Check out the QR code. Go to these articles if this is of interest to you. One of the things that we see is in animal models, this brain clearance does contribute to the clearance of amyloid. It does contribute to the clearance of tau. It does contribute to the clearance of alpha-synuclein. And in animal models, if we impair this clearance, because we have different ways we can impair it. There's drugs we can use genetic manipulation, right? We can use surgical manipulations. It does promote amyloid pathology. It does promote tau pathology, and it does promote synuclein pathology. Don't take my word for it. So I'm going to we're going to nerd out a little bit just to get you to this data, but I'm going to show you one of these threads. So my lab studies this molecule called aquaporin 4. There will not be a test on this. You don't really have to remember it. It probably doesn't matter that much in your day-to-day life. We care a lot about it because we think it's really important. Aquaporin 4 is a water channel. It allows water to move in and out of cells. If I look into the brain, this is of a rat, and I look at where the aquaporin 4 is, you can see it's labeled here in purple. It looks like it's along blood vessels, right? It's not in blood vessels. It's actually it's on a part of the cell. These star-shaped cells are called astrocytes, and they actually ensheath, they wrap up the blood vessels. So if I'm a blood vessel looking out of the brain, almost the only thing I see are astrocytes, and almost all of that surface area facing the blood vessels is made up of this water channel. What we think this water channel is doing is it's organizing water movement through brain tissue into and out of these spaces around blood vessels. So it's organizing fluid movement around these vessels, right? We think that's how it gets that organization, is because of this channel. You can genetically manipulate mice to get rid of the gene that makes this protein, and you get less movement of fluid into brain tissue. And if we measure the clearance of radiolabeled amyloid in an animal that has normal amounts of aquaporin 4, about 70% of the amyloid's cleared in the first hour. In the animals without this protein, it's slower. So this water channel is a really important part of this whole clearance process. I bring this up because this is our tool. We know that this water channel is important, so if we disable this water channel, we disable brain clearance. And that's the tool we use to ask, if I disable clearance, do we get more amyloid? If I disable clearance, do I get more tau? If I disable clearance, do I get more synuclein? If you've ever been interested in mouse husbandry, this is your opportunity. So the nice thing about mice is they can be genetically manipulated. We have genetically modified mice that make boatloads of amyloid beta. They get plaques. They have plaques even in young adulthood, right? So they have very aggressive amyloid pathology. And we can cross them with the mice that don't have aquaporin 4. So these are mice with a disabled clearance system. And the question is, if I disable the clearance system, do I get more plaques in those mice? And if I sort of look here, plaques are labeled in red. These are in the animals with aquaporin 4. These are the animals without. There's more red on the right. We can quantify that here. So if I disable this clearance process, I do get more amyloid. And the same is true of tau, and the same is true of alpha-synuclein. I'm not going to show you that. Okay, so then, what about people? What about human Alzheimer's disease? Is this what's happening in Alzheimer's disease? Is aquaporin 4 going away? Is that the problem? Do you need more aquaporin 4? Do I need more aquaporin 4? The answer is not so simple. It's never that simple. We have an idea, and then it's always wrong, and then you have to come up with another idea. It turns out that as animals get older, they actually get more aquaporin 4. But it's not where it's supposed to be. So this is in a healthy young animal. It's kind of washed out here, but you can see almost all the purple is along this blood vessel. This is one astrocyte, that's one capillary. You can see almost all the aquaporin 4 is right at that blood vessel where it's supposed to be. In the old animal, look at this cloud of purple. It's everywhere else. So if you think of this, if this cell's job is to route water only into and out of this vessel space, this one can do it, this one can't. Because this one, water's just going to go everywhere. And so what we think is happening is there's a loss in the localization. The placement of this channel is actually a problem. We see this in aging. We also see it in brain injury. We also see it in people. So this is a study we did in collaboration with the Alzheimer Center down in Oregon, where these are 60 people, 65 years of age and older. Some of them, these are people who've passed away who've donated their brain to the Alzheimer Center for us to study. So these are cases who were cognitively normal, some who had mild cognitive impairment, and some who had Alzheimer's disease. Long story short, if we measure how perivascular the aquaporin 4 is, we see that people with Alzheimer's disease, they have less localization. If we look at localization, like where the aquaporin 4 is, so if it's in the wrong place, that's over to the left, the wrong place, it's over to the left, we see that worse placing is associated with worse cognitive decline and worse amyloid and tau pathologies. This is a little bit nerdy, so you don't have to go too deep into this. These are two different measures of cognitive and functional decline. This is a measure of amyloid pathology. This is a measure of tau pathology. What you can see is less aquaporin 4 in the right place, more pathology, worse cognitive function. So in people, even people without Alzheimer's disease, changes in aquaporin 4 do in fact associate with cognitive impairment and Alzheimer's pathology. So if what happens in the aging and the injured brain is aquaporin 4 goes to the wrong place, is that what can that cause more amyloid, more tau? Well, we have a mouse for that. We have a mouse for everything. That's what's great about mice. We know that aquaporin 4, it sits where it sits because it connects to a string of other proteins. And if we delete the gene that makes each of these proteins, we get this. So normally you get aquaporin 4 that's all along blood vessels, like here. If I delete this yellow gene, I get aquaporin 4, I get normal levels of aquaporin 4, but it's just everywhere. So this is very similar to what we see in the aging brain and in the injured brain. More mouse husbandry. I know you were hoping for that. Here it is. You're welcome. So when we cross the mice that have aquaporin 4 in the wrong place with the animals that make a bunch of plaques, lo and behold, the animals that don't have aquaporin 4 in the right place, tons and tons and tons of plaques. So, is it at the scene of the crime? It does seem to be. Does it have the means and the motive? It seems like impairment of this process does seem capable of promoting this kind of pathology. And so now this is when we do the grand reveal, when I show you the killer experiment that we did that shows that if we enhance glymphatic function, we actually cure Alzheimer's disease. We haven't done that yet. That's where we're at. So over the past 12 years, the data suggest they're consistent with, they support a role for an impairment of this brain waste clearance in the development of Alzheimer's disease. But the clear causal role, right? The fact that I could intervene and change the fate of a person who was gonna get Alzheimer's disease, we're not there yet. That study hasn't been done. We're trying to design those things right now. This is a work in progress. Why do we care? Why does this matter? Let's bring this back up. The Jack curve. Remember, the cognitive impairment is the last thing that happens. The development of amyloid and tau pathology happens decades before. Remember that removing amyloid, it may offer some hope. That's where the current therapeutics are aiming at. They're trying to get rid of amyloid. Some future ones will probably try to get rid of tau. That's good, because if you've already got amyloid in your brain, you probably want to do something about that, or you at least want the ability to do something about that, right? But at least, it's actually, frankly, it's probably too late for me. At least for my kids, I would love for them to have the ability not to get the amyloid in the brain, right? And for us to really tackle and defeat Alzheimer's disease in the same way that we've made a lot of progress in the setting of heart disease, that's what it's going to take. It's going to take identification of people who are at risk of developing amyloid and treating them before they get it. So the interesting thing is, what these data suggest is that glymphatic impairment and sleep disruption, it's to the left. It's at least one of the things that appears to be driving these two processes, right? So if we had a way, an MRI scan, a blood test, a device that could measure this impairment, we could identify not just people who have amyloid plaques, we could identify people who are at risk of getting them, right? That's important, right? If we're going to prevent things, we have to know who's at risk, because we can't just give everyone the drug, right? That doesn't work. If we can figure out how this process works, if we can figure out exactly how it goes wrong, we can develop a drug, a behavioral intervention, a device, right, that restores or rescues this impairment, thus preventing these processes. And this is how we would go about the process of Alzheimer's disease primary prevention, targeting this process. That's where, at least for me, that's where I hope we're going. All right, so take-home messages. I often get questions like, so what does this mean I should do right now? Unless you're a mouse or a rat, this doesn't necessarily say you should do anything yet. Right? This is early science. This is early days for us. So in terms of actionable conclusions, I'm not sure we're there. But it does tell us a couple of things. It suggests sleep's really important. And we already know that, right? I think everyone understands that intuitively. But when we're thinking about cognition and dementia, sleep might be a modifiable risk factor. So if you have obstructive sleep apnea, if you have chronic insomnia, right? Might be wise to think about that. If you're a 40-year-old professional who sleeps 4 hours a night and you think that, oh, don't worry about it, I'll sleep when I'm retired, It's midlife sleep disruption that is the risk factor, not late life. So what that suggests is the sleep that we're getting in our 40s and in our 50s is actually setting the stage for what our brains are going to be like in your 60s and your 70s. That matters. So treating things that need to be treated. There's also this other thing. One of the things that we see is cerebrovascular disease, traumatic brain injury, sleep disruption. All of these risk factors all seem to converge on this biology. It's likely that many of us have different risk profiles. Some of you might be old. Some of you might have had a whole bunch of concussions. Some of you might have had a major brain injury. Some of you might have heart disease. Some of you might have high blood pressure. Some of you might have a genetic predisposition for some of these conditions. All of these different contributors are all sort of adding together to drive your risk. So if you have 1 or 2 or 3 of those risks, I don't know, if that's me, I'm probably going to do whatever I can to mitigate the other risks that I can do something about. And sleep is frequently one of the things that we can do something about. So I think that provides some rationale there. More importantly, what I hope to convey, I was trying to sort of lift the hood a little bit on the discovery process. What a neuroscientist is doing? How do we discover new things? How do we evaluate its importance? Neuroscience, sleep science, Alzheimer's disease research, and it's moving fast, and it is exciting. While there's no, hey, I've got a drug I'm going to sell you, that's not a part of this talk, and it probably won't be a part of a talk for another maybe 5 or 10 years, right? I probably will never be selling the drug, but someone might be. But things are moving very quickly right now, and there's a lot of interest. There's a lot of funding, and frankly, threads are starting to kind of come out that people are starting to pull on, and it's starting to be real productive. So I think that this space is going to change a lot over the next 10 years. So I think it's definitely something to watch and something exciting. So this is where we're at. It's to be continued, right? So it's an incomplete story, which science is. I hope it was interesting. I hope you didn't fall asleep. I saw some of you did fall asleep. Good for you. So thank you for your attention. This is my lab who does all the work. These are our funders. And thanks. If you want to talk to me or do anything like that, this will take you to my lab's website. Appreciate it.
That was awesome. And I just want to thank Jeff for giving that talk, giving us some hope, giving us an overview of the science, and really providing us a little bit of a primer of what the scientific process looks like. So what we'd like to do now is take some of your questions. And I'd like to have Jeff come and take a seat up here along with Don Howard. Don is an old friend of mine who is a sleep medicine doc here in Spokane. He has worked for many years in the ICU and pulmonary and now is doing exclusively sleep medicine and is just one of the smartest folks I know. So, not too much pressure there, Don. I'm going to try something new tonight, and that's when I go home, I'm going to say, honey, I'm going to go wash my brain. And see what kind of response I get. So I think we have a mic. So do we have an extra mic? If you have questions, raise your hand and we'll try and get to those. I'm going to start off with one question that I wonder about being a doctor and being up many, many, many nights of my career. Is there any going back? Jeff? Don?
So the question is, how much bad behavior is too much, right? is what you're asking. We just don't know. There's here in Spokane. There's a really great research group that does work on shift work. It is many of our suspicions that's going to be a big risk factor for some of this. We don't know an answer to whether this is recoverable. The data that when people have looked at a single night of sleep deprivation, they see that the effects, including the buildup of amyloid that occurs with one night of sleep deprivation, it takes days to clear. And so if we're talking about, if you were, on call overnight for a decade or two of your life, I don't know, maybe long-term care insurance. Is something you should think about.
And
but
the
study is the problem is we haven't done the studies with sufficient resolution to answer that kind of a granular question yet. So we don't know.
Don, do you have thoughts?
I feel like I'm in trouble. No, I think from a clinical standpoint, number one, I thought that was a fantastic lecture. I actually learned a lot. But just getting people to understand more about sleep and trying to get people to understand they need to get sleep in the clinical setting is extremely hard. I can't get people to change habits, to have a set bedtime, a set wake up time to get enough sleep. And you promote it, but I think the majority of us, that just go to sleep every night are kind of thinking, it's just something you do, but it doesn't play a big role in your health overall. But if you're a normal sleeper, like you don't have sleep apnea or some other medical problem at all, and you have normal sleep, if you can just spend 8.5 hours in bed to get 8 hours of sleep, roughly, you're doing yourself a huge favor. That's what I tell patients every day. But everyone has a reason, right, why they can't. And a lot of it's their favorite TV shows on around 10 p.m., so they can't get to sleep. Or setting a bedtime, setting a wake-up time. That's really, I tell people all the time, the worst sleepers are teenagers, retirees, disabled, and people who live alone. And mainly it's because they don't have the schedule they had when they were working. When you're working, you have to get up at the same time every day. If you get up at the same time every day, you tend to go to bed at the same time every day. If you live alone, you don't have someone telling you to go to bed. If you're married or have a partner, then you tend to have the same schedule and you go to bed together and you kind of drive each other to do that. Teenagers are just teenagers. It's going to be what it is. They probably get enough sleep. They just don't have much of a circadian rhythm. So I think just from a clinical standpoint, it's promoting, good, what we call sleep hygiene, good sleep practices.
I think we had a question down here in front.
Yeah. I don't know.
Use your outside voice. Yeah.
So I have type 2 narcolepsy. Dr. Howard actually was the one who did my sleep study and gave me the diagnosis. I have tried, I have done sleep hygiene. I have done an online course in cognitive behavioral therapy for insomnia. I don't sleep. Some nights I sleep, most nights I don't. I feel desperate. I do use, fortunately, I can get by with just a very tiny little bit of generic Ambien, but I don't like using it. What do I do?
Well,
Don, can you repeat the question or summarize the question?
Yeah, the question is in type 2 narcolepsy, which is narcolepsy without cataplexy, type 1 is narcolepsy with cataplexy, is that she's complaining that she doesn't sleep at night, which a lot of you might think with narcolepsy doesn't make sense, that they sleep all the time.
And I want to mention that I used to sleep really well until I went through menopause.
Yeah.
And then it was all over.
So one of the things about narcolepsy is that it is commonly associated with insomnia and a lot of sleep fragmentation. So probably 50%, I don't know if that's an exact correct answer, but 50% of my patients that have narcolepsy complain of very fragmented sleep, interrupted sleep, a lot of awakening is at night, and they think if they just got a good night's sleep, then they would feel good the next day. But it doesn't work that way. You can improve their sleep, and they're still going to be sleepy because they're missing something in their brain called hypocretin cells. But in your case, insomnia is a huge problem and question, but just from a narcoleptic standpoint, there are medications out there now called sodium oxybate that we're now using in narcoleptics to improve sleep at night. And it improves their symptoms during the daytime. But that's a very, very common symptom in narcolepsy. So we should be able to make that better. Okay.
I have a question in the back, and then we're going to come back to the front and go to the middle.
My name is Gil Smith. I'm a retired OBGYN. So by definition, I'm sleep deprived in my 20s, 30s, 40s, 50s, even early 60s. So I'm frequently up in the middle of the night every second or third night. So I feel like I'm doomed. I don't have a genetic history of this problem. And even now I get up in the middle of the night for different reasons. But I take a nap in the afternoon. I just wonder if that's helpful. I don't feel like I'm in Alzheimer's stage, but
Yeah, just a little bit of epi data. So First, we should actually start a study on brain donation for former OBGYNs, because I think it would be super interesting. Not to speed that along or anything like that in your particular case, but it'd be really interesting. So napping is a question people ask a lot about. The epidemiological data on napping is a little bit mixed. Some data suggests that people who nap are at an increased risk of cognitive impairment. And some data suggests that some people are at a decreased risk. When you read what scientists, when scientists get data that goes two different directions, we're forced to make up reasons why that's true. And sometimes those reasons are plausible and sometimes they're just not. My thinking, and I think some people's thinking on that is, the reason for napping matters. So if you're a terrible sleeper and you're sleeping, napping during the day because you're sort of doing it out of this need or out of this deficit, that's probably not a good thing, right? And that probably underlies some of that risk behavior. On the other hand, if you have a very stable sleep schedule and you have a very stable sleep diet and it happens to include regular napping, that's probably a positive protective thing, right? So I think it a little bit comes down to why you're doing it, whether it might be protective. But the jury's still a little bit out on napping. I like napping. I nap at my office.
I'm a nap enthusiast
What I would say to
myself.
that is that, so we all have a circadian rhythm, which is our body clock, right? Tells us when to be awake, when to sleep. And, supposedly we're supposed to, whenever, say you go to bed at 10, that's when all of your melatonin's going up and your temperature's dropping and all these other things are happening to prepare you to sleep. And your cortisol's low and it stays low all night long. And then before you get up, your cortisol level starts going up and your temperature starts going up and you're exposed to sunlight and your melatonin drops and you're supposed to be awake all day. Well, that same thing, right around between noon and 3, everyone has something called the circadian rhythm dip, where they get drowsy. Everyone's is deeper. Mine's pretty deep. I mean, I feel it. I feel like I want that to close my eyes. My wife's is very shallow. She doesn't feel it much at all. So I tell patients when I see them that napping is fine, but you should never nap longer than 45 minutes. We call these kind of power naps. If you sleep too long, you will get into stage 3 sleep, and then you wake up feeling groggy and sometimes irritable, and it can affect your sleep at night. So power naps 30 to 45 minutes, somewhere between noon and 2. From a clinical standpoint, we say is fine because we all feel that. It may not be due to you have bad sleep at night. But if you're sitting and sleepy throughout the day, and you're wanting to nap, then I would agree you should probably get that figured out. Why are you feeling that way if you're spending enough time in bed?
So I want to do due diligence to our online audience. I got a question from the online audience. And that was, I know that you don't have human data on this likely, but in terms of the lymphatics and washing your brain, what effect might you have on that from pharmacologically induced sleep in an animal? So you give an animal a drug to help them sleep. Does this process change?
Yeah. So one of the nice things about the animal studies is we can give animals all sorts of drugs and ask this question. One of the more important studies in our field that was done, it was this very heroic study by someone, her name's Lauren Hablitz, where she gave animals 8 different sedative agents. Sedative agents, so like anesthetic agents or the things you would take for sedation if you were undergoing some sort of procedure, right? They do different things. And they induce different electrophysiological states within the brain, and they have different effects on your cardiovascular system, right? And what that study showed was that some agents dramatically suppressed brain clearance, and some agents dramatically enhanced it, right? These are all sedating agents. These aren't agents that are being used as sleep aids, at least not at this time, right? What we can infer from that is the types of drugs that you might be taking for sleep are probably going to have an effect on this biology, and it is probably going to connect to what kinds of sleep-like electrical activity those drugs produce. Right, so the example that I think of is the drug dexmedetomidine, which is used in sedation. In animals, dexmedetomidine dramatically enhances this process. Electrophysiologically, if you look at what dex does, is it creates very profound, slow wave, low frequency activity that is very similar to what, in fact, it's a little bit slower than what you see in N3 sleep, in slow wave sleep. Right? So we think that one of the reasons why it does that is because it's enhancing the same thing that is present in slow wave sleep. So I think our sleep physician should probably comment on what types of sleep aids produce.
Thank you. No, it's so getting real quick into insomnia, which is a whole discussion in itself, right? Do you use medication to help with insomnia? Do you not use medication? And mostly physicians would recommend trying to avoid sleep medication if possible. I mean, we don't want to use anything if we don't have to. So cognitive behavioral therapy, as someone discussed earlier, is still kind of first-line therapy, which is a training way of helping you sleep without medication. If we're going to use a medication, the problem now is there's so much data coming out regarding certain medications that we use for sleep are potentially associated with Alzheimer's, and these are called anticholinergic medications. Specific like amitriptyline is one. Over-the-counter Benadryl, you should not be using over-the-counter Benadryl. We don't recommend that anymore. It's in everything you take over-the-counter pretty much. There's Seroquel is another one, Doxepin is another one. There's like 6 to 8 of them. So we're kind of in a rock and a hard place of what to use in someone who's not responding to like cognitive behavioral therapy. If they're not sleeping much at all, what do you use to try to enhance their sleep? So in my practice, I won't prescribe any medication until they show me they can maintain a sleep schedule. If they maintain a sleep schedule and do all the cognitive behavioral therapy stuff that we have talked about over quite a while, at least 6 months or more, and they're still not sleeping, then we have a discussion about medication. But then you get into the Ambiens, which everyone's scared of, right, for unknown reasons, but everyone's totally scared of Ambien and Lunesta and some of these GABA agents, and benzodiazepines we like to avoid because of addictives and stuff like that. And then you look at the old medicines we used to use, and these are the ones that are saying that they have anticholinergic properties that potentially worsen Alzheimer's. So insomnia is hard, and getting people to change their habits is the biggest thing we like to do for insomnia, and try to avoid medication as much as possible. But there are times that we do, and we have a discussion and determine what we're gonna treat them with.
And Jeff, if I heard you right, you said that the effect of the medications kind of depends on the exact role of the medication, how it's inducing sleep on the glymphatic system.
Yeah, the work that has been done, it seems like the effect of the drug on the glymphatic system seems to relate most closely to what kinds of electrophysiological activity it induces. And all of these drugs, many of these drugs, induce certain types of activity, right? But we and others are actively evaluating some drugs in this space, so we'll find out.
Yeah, science. I'm gonna go to the front row, then we're gonna go to the middle, we're gonna go to the back, then we'll come back here. So.
My question is sleep gummies. What's your thoughts on that? It's like CBN.
I'm actually would be pro.
What was the question? Yeah.
CBNs. And now I just
So what CBMs are?
yeah. CBN, right? Right. I just blanked on the name, so say it. Cannabinoids. So the reason I pro on it, and apparently I have Alzheimer's as well. But there's some stuff coming out now that these agents can be very effective, even with a little THC as well, as far as sleep is concerned, without potentially the same side effects. I mean, I think we're finding that cannabinoids in general are becoming used in a lot of ways, but we have this Ronald Reagan fear of them, and Nancy Reagan just say no, like they're a starter drug and all this.
Really well, but it's a good sleep.
Oh, that's okay. I can answer that question. Okay, that's a very good question. The question was, she sleeps well on her CBNs that she's taking, which are the cannabinoids. It's like, it's not THC, but it's one of the products that's, and the cannabinoids are. You are taking half THC. So these agents aren't made for sleep, but they cause relaxation, they can cause drowsiness, they can help sleep. But I want everyone to understand, when you take a medication to help you sleep at night, it doesn't improve your sleep architecture. In other words, you have sleep architecture, stage 1 sleep, stage 2 sleep, stage 3 sleep, and REM sleep. We all have very similar percentages of those if it's not being interrupted by like sleep apnea or something else. Like you have roughly 10% stage 1 sleep. You have roughly, these are rough numbers, 60% stage 2 sleep. You have roughly 15% stage 3 sleep and 15% REM. Those are rough numbers I give patients because they all have their Apple Watches. Okay, and they're bringing them in every day. And so I kind of go over them with them and they say, my Apple Watch, if you look at it, it uses the word light sleep. There is no such thing as light sleep. There's just sleep and different stages. Light sleep is stage 2 sleep. And so they're seeing all this light sleep making up like 60% of their night. They're going like, I'm not getting any deep sleep. I'm like, that's because it only is going to make up 15%, maybe 20% depending on your age. If you're little kids have huge amount of stage 3 sleep. And as we get older, we have less. So medications don't give you more of a certain stage, but they do allow you to stay asleep longer. So if you are asleep longer, you would have hopefully more stage 3 sleep where a lot of this glymphatic function is occurring.
You wouldn't say it's damaged.
I don't think I think most of the things coming out are saying it's much safer than most of the things we're probably prescribing.
All right, we have one question back in the middle.
Actually, I've got two, if I could, please, and then I'll be done. So typically, if one has sleep apnea, are they waking themselves up gasping for air through the night necessarily?
Well, sleep apnea, people say a lot of times I stop breathing in my sleep, and I tell everyone no one stops breathing in their sleep. Apnea, the best way I can explain it is like breathing through a straw, okay? And I pinch the straw. You're still moving your chest. Because breathing is moving your chest. The action of actually moving. Not breathing is like pausing, holding your breath. That sleep apnea is not holding your breath.
No.
It's your airway closing,
Right.
and then your effort to breathe actually sets, when you hit a certain negative pressure in your chest, trying to breathe is what wakes your brain up, which then sends a signal down to open the airway. So every time you have an apnea, there's an arousal associated with it. So that fragments your sleep. And that's going to decrease the amount of stage 3 sleep you get. It's going to decrease the amount of REM sleep you get. It's going to make you sleepy. It's going to give you poor sleep quality.
Okay, thank you. And then the other one was, I do detailed dreams pretty much every night. Is that a good thing?
I have no idea. Dreams are those things, right? That it's just so.
Okay.
In clinic I limit the dream questions because that could go on for a long time. You only have so much time.
Because I
But
thought
will
you
tell
mentioned it's in REM when you're doing the dreaming.
You can dream actually in any stage, but
Oh,
like
okay.
most of it, probably 90% of it occurs out of REM. But you can dream in other stages. But the thing about dreams is there are certain conditions, like, for example, narcoleptics tend to have very, very vivid dreams. People with PTSD can have nightmares related to that. So dreaming, like there was a thing that came out and it said that we don't dream about what's going on in our life. And I was like, that's not true. I dream all the time about what's going on in my life. So I never really understood that. And every patient I ever meet tells me they have dreams about things that's going on, But to try to figure that whole dream thing and what it's doing, but it's felt to be like stimulating neurons, which then give you better connections, and in the young brain, REM sleep is much more prevalent than it is as we get older.
All
I have
right,
a question up here. I have really enjoyed this. I've learned a lot. And I appreciate your telling us that there are factors in our life that we can change to make this not as prevalent. But you haven't talked about the genetics. And what does that mean?
So you're talking about the genetics of Alzheimer's disease.
So there's a couple of different genetic angles to think about, right? So Alzheimer's disease itself has a strong genetic element to it. There's a small proportion of Alzheimer's disease is a familial early onset form, but that's a very small proportion of cases. The vast majority of cases are what is called sporadic Alzheimer's disease. which is typically hitting people over the age of 65. But that, even though it isn't, we don't call that a genetic disease, right? So the familial ones are actually caused by a very specific gene mutation that always causes the condition. Sporadic Alzheimer's disease is influenced by a number of genes, the strongest of which is the ApoE gene. There's a specific variant, the E4 variant, that drives a lot of the risk in the Alzheimer's disease space. So that's when you do 23andMe, that's the thing that they're reading out that defines a lot of your Alzheimer's risk. There are also genetics of sleep. There are people, so groups have done genetic studies on people and their sleep performance, and there are families of people that have short sleep, or that tend to have short sleep, and there are families of people who tend to have long sleep. Morningness and eveningness has a genetic component to it, does it not?
Yeah.
So there's definitely a genetic element to sleep and how it goes. How that modifies the relationship between sleep disruption and Alzheimer's disease and dementia, I think, is largely unexplored. This glymphatic function stuff that I was talking about today, this brainwashing, some very early studies have started to ask questions about all the genes I talked about today, so aquaporin 4 and alpha-syntrophin and distrobrevin, these genes that are part of this biology. There does seem to be a connection. So we've done a study out of a big national Alzheimer's study where we do see that naturally occurring variants in some of these pieces of this process do seem to associate with either more rapid or slower cognitive impairment, right? So there might be a genetic component to this biology, right? Separate from the genetics of sleep, separate from the genetics of sporadic Alzheimer's disease.
All right, we have one more in the middle here.
Is there any research being done on this glymphatic function in meditation? So not just sleep or awake, but in states of meditation?
Yes. So we have a grant. We're studying it in Tai Chi practitioners. Tai Chi is not strictly a meditative practice, but it's a mindful movement practice. I'm aware of someone, and I know that neither of these things that I'm mentioning are meditation, but there's a group in Oregon that's looking at yogic breath work and its effect on this. So from the perspective of, so there could be direct effects of meditation on this biology that go through autonomic functions, so your sympathetic arousal, and the electrophysiological states that your brain arrives at during meditation. Right? There's also likely secondary effects, and this is what we're expecting in our Tai Chi practitioners. These are people who practice Tai Chi sleep better. They're fit. They are leaner. They tend to have better cardiovascular health. All of those things are things that we would expect to improve this biology, right? And so while your glymphatic function may not be higher when you're doing Tai Chi, people who engage in those kinds of activities, the same way a person who is who exercises regularly and has good cardiovascular fitness, we would expect those people to have more function. But people are definitely in that mindful space in the glymphatic field now. It's hard to get a mouse to meditate, though.
That's what I was going to ask you, if you've taught a mouse to meditate yet. We have a question down here.
My question is, eating at night. Should you shut off your eating? How many hours before you would? What happens if you eat all the way after bed?
I mean, these are all kind of what we call sleep hygiene, practices. Are they the same thing for everybody? And the answer really is, no. I mean, some people can do anything and they're going to sleep well, right? They could literally, drink a 6-pack, smoke a cigarette, and take a stimulant and drink a cup of coffee, and they would still go to sleep and sleep all night. They're just good sleepers. But if you have insomnia, where you don't sleep well at night, there's just a lot of general rules. And, in general, for no caffeine afternoon, right? Because it has a pretty long half-life. So you want that out of your system by then. Exercise actually in the late afternoon is better for sleep because you don't want to exercise before you go to sleep because your temperature is going up when you're trying to go to sleep and it should be coming down. But if you exercise between 4 and 6 and you're going to bed, let's say at 10, your temperatures rise and now it's falling further, So these are some things that can help. As far as eating, large meals, we roughly say 4 hours. If you're going to have a snack, that's okay. So it's really kind of dependent. And I want people to understand that if you're already a good sleeper, it's not like by doing these things you're going to have better sleep waves. You're just going to have sleep waves. But yeah, those are the kind of general rules. I think the biggest one is really getting people to understand the circadian rhythm is 25 hours long. Your body clock is 25 hours long. So if I put you in a room that has the same light and a bed, and you can get food and you can get anything you want, but you have no idea what time it is, you will go to bed 1 hour later and get up 1 hour later every night. Until pretty soon you're going to bed at 10 a.m. when you think you're going to bed at 10 p.m. And that's called the free running circadian rhythm, okay? The way you fix that is if you get up at the same time every day, then you're restarting your day at 24 hours instead of 25. So the wake-up time is a lot more important than the bedtime, with the exception of you need to get enough sleep. So I tell people to pick a wake-up time and say, just like you're working, you should get up at this time every day. Now, no one's going to totally do that, but if you get up the same time every day, then go back 8.5 hours, that's where you should go to bed.
I think this is all just so fascinating. We all sleep, and how much we know and don't know about sleep. I'm sorry, Don, I interrupted you. You have something else to say? We have one more question back here.
Yes, I want to thank you for your presentation tonight, Dr. Cliff. I'm actually a patient of Dr. Howard, and I had an appointment just this morning.
I already told him,
And I'm a patient who underwent 3 sleep studies for obstructive sleep apnea. And when I read your article in the paper about the glymphatic system and its impairment, my question is, does an impaired glymphatic system have a negative effect on glucose metabolism? And those who eventually have, type 2 diabetes?
So I am not aware of any data that has looked at that question. If you ask a scientist a question like that, though, and they don't have an answer, they're likely to start making stuff up. So I'm gonna make up 2 things. So one is there is a little bit of data in animal models to suggest in the animal models of type 2 diabetes, you see glymphatic impairment. So you see it running the other direction. So in the presence of induced type 2 diabetes, you can give a mouse that or a rat that, you do see an impairment of this process, right? Whether that's running through the vascular dysfunction that you see in that setting, that's likely, or there could be other reasons for that. More generally, we have this biology has been billed over the last 10 years as waste clearance, and we've been very focused on Alzheimer's disease and amyloid and tau. But the same biology also moves things around and spreads things around. So like in your brain, you get inflammatory molecules that arise as a result of some sort of insult, whether it's an infection or an injury or something like that, you get inflammatory molecules. Well, those have to get cleared, right? Or those need to move to different places. Well, the same process that clears stuff away also distributes them, right? So when we think about the possible clinical implications of this biology, we actually think they're pretty far-reaching. We think that things like norepinephrine and serotonin and dopamine, which in the brain are released sort of in a cloud, like it's called volume transmission. They're not released synapse to synapse, like connection to connection. It's like they're released into the swimming pool, right? And the dynamics of those molecules in the brain, in the spaces between the brain cells, are almost surely being regulated by this fluid movement. And so we are actively starting up studies looking at whether this dysfunction is related to mood disorders, bipolar disorder, PTSD, all sorts of things. So when you talk about like metabolic regulation, could there be an arm going that way that has to do with the kind of the neurobiology of brain signaling? Sure. But that's a made-up answer.
Thank you very much. And thank you, Dr. Howard, for your exceptional care. I appreciate that.
So I know you had one other question regarding diabetes and sleep apnea as well. And there is a correlation between sleep apnea and type 2 diabetes with regards to insulin resistance. Felt to be mainly related to the sympathetic stimulation of sleep apnea, like when you have an apneic event and your brain wakes up to basically open your airway. It's like a sympathetic, like a fight or flight response. So there's an increase in what we call sympathetic tone of your autonomic nervous system. And that increased tone actually affects insulin resistance. So it does actually affect, glucose controls is the best way I guess I can put it. So there is that kind of correlation between that. I'm not saying I'm reading every research article out there with practicing every day, but that's what I can tell you about that.
All right, we have one last question in the back. And
Yeah, I was just wondering if there's any herbs and minerals that are helpful with sleep, like, certain types of magnesium, valerian, those types of things.
That or me. Yeah, I mean, there's a bunch of them out there. It's difficult when you practice sleep medicine because what you see is the worst. You don't see the people who have mild conditions. Like if you have a little bit of an insomnia, occasionally here or there, or you just think your sleep's not as good, there's a lot of kind of natural sleep aids, and there's a lot of things that are mixed multiple, into one. And these are, your melatonins, which if you look at melatonin, it's never really been shown clinically to be that effective for insomnia. But I see it every day where patients tell me it helps them. I'm certainly not going to argue that fact. But valerian root is definitely one. L-leucine is another one. Kava kava. There's I'm trying to think of some others that are in these different meds. GABA. So if you kind of look at, I did this the other day just because I get a lot of questions about it. Just go online and just Google best over-the-counter natural sleep aids. Just do it. You'll come up with most of these products that you're going to see are something you're going to order online. They're probably not going to be cheap. But most of them have all the just natural, sleep aid stuff in them. And the reason I'm starting to bring this up more is that I feel like from a standpoint of prescribing medication, we're kind of running out of options based on the effects of medications with regard to brain function and cognitive function and things like that. We don't want to use anything, but people don't come to you with insomnia because their primary could fix it. They come to you with insomnia because no one's been able to fix it. And so sometimes you do have to use meds. So I think the answer is, I think there's some things out there.
so Don, I'm gonna, you're bringing out the doctor in me, sorry. The only caveat I would add though is if you're taking a supplement, we don't know what it is. We oftentimes don't know the strength of it. It's not very well regulated. And we don't know how it interacts with other medications you're taking and whether it's gonna dump you into a toxic range or increase the metabolism of that med and make it ineffective. So what are your response to that?
Well, yeah, I think whenever you come up with supplements, that's kind of where you're at, right? But, sleep medicine is so different than like pulmonary, what I practice in critical care, which is kind of like, you're just trying to save the life. You're not trying to think how long they're going to live. And in pulmonary medicine, you're dealing a lot with asthmatics and smoking, things like that. Sleep medicine is so kind of touchy-feely. There's such a relationship between you and the patient because they sleep and you sleep, so they understand what sleep is. And they just taking the history, and there's so much conversation with a sleep patient about what they think as well. What do they want to do? How do they sleep? Can they do this? Can they do that? Where normally as a physician, you're telling something, you need to do this. You need to quit smoking. You need to quit drinking. You need to sleep more. But there's a lot of this conversation that goes on. And when it comes to like insomnia, which every sleep doc, it's the worst for us. It's the hardest thing to treat, because there's so much back and forth. The patient, I can't do this. No, but can't do that. Can't do that. Can't do that. My husband won't allow me to do that. Can you get the TV out of the room? No, my husband won't allow me to get that. He likes the TV on all night. And I'm like, well, you're not going to sleep. Well, your husband snores really loud. Yeah, but he won't get a sleep study. I mean, this is, and so I'm like, well, what do you want me to do? You know, you won't get rid of the TV. Your phone's on. Your husband's snoring. He won't get a sleep study. Your husband has 2 dogs. He loves the dogs. They sleep in the bed with you. Your grandkids visit every weekend, and they come in your bed with you. And you're there telling me you can't sleep, and at the same point, you're telling me you can't do anything to change it. That is the biggest conversation when it comes to insomnia. All other sleep disorders like narcolepsy, which I've said this before, I love treating narcolepsy because you can make such a big change. Insomnia, actually, you can, but it's difficult. We need more cognitive behavioral therapists, and we don't have that. That's what we need.
I just heard Jeff broadcasting his thoughts about he loves his mice. So I always want to say thank you so much for coming tonight. And can you please give both Dr. Iliff and Don Also, just before we close, I want to thank our sponsors. Again, the Spokane Regional Health District, Spokane County Medical Society, and UW Population Health Initiative. This lecture has been recorded, so in about a week or two, you'll get an email with a recording. Please feel free to share. And once again, just thank you for coming tonight, showing support for not only our partnership, our community, but such great community activists and great researchers on our behalf. So thank you so much.